Welcome to the deep dive. We're taking a close look today at some really exciting preclinical work, stuff presented at the recent ATS 2025, and also drawing from LOMJ 44.
That's right.
Our focus is the potential of inhaled RNAi therapeutics. These are from Arrowhead Pharmaceuticals, looking at tackling some major lung diseases.
Exactly. We're diving deep into how targeting specific inflammatory drivers in the lungs, think of molecules like IL-33, TSLP, and RAGE, how targeting these with this, innovative RNA interference approach could really change the game.
For which conditions specifically?
For things like asthma, COPD, and even ARDS. Our mission today is to really understand how these strategies could potentially lead to more effective treatments, treatments that get closer to the root causes.
Yeah, and what really caught my eye was the promise of treatments that might work more broadly, more effectively maybe than what we currently have.
Yeah.
We're talking about potentially overcoming some limitations of current therapies for these, well, very common and often debilitating lung conditions. Okay, so let's start maybe by unpacking this dual targeting approach for allergic asthma. This works as highlight that one.
Yeah, that's a great place to start. What's fascinating here is the focus on IL-33 and TSLP. Now, the sources call these upstream alarmins.
Alarmins, like warning signals.
Pretty much. Think of them as early warnings released by airway cells when they're stressed or damaged. And what's really significant is they're involved in not just the classic type 2, allergic inflammation and asthma, but potentially in other inflammatory pathways too. So it suggests a broader impact than just, say, one specific type of asthma.
Okay. So when we talk asthma, it's not always just one simple inflammatory picture.
Precisely. These alarmers, they can trigger a whole cascade of immune responses involving various cell types, leading ultimately to those symptoms we associate with asthma. So the ATS poster, it details a preclinical study in rats where Arrowhead used their TRiM platform to deliver a single, what they call a heterodimeric RNAi.
Heterodimeric, so 2 in 1.
Basically, yeah. It's a single molecule engineered to silence 2 different genes at once. In this specific case, both IL-33 and TSLP.
Huh. That's a pretty clever way to, get more bang for your buck at the molecular level, and it sounds like it actually worked.
It did, yeah. The study showed this dual-targeting RNAi was just as effective at knocking down both IL-33 and TSLP levels as using 2 separate RNAs would have been.
Okay, so feasibility confirmed.
Exactly. And even more exciting, they saw additive anti-inflammatory effects.
Additive, meaning?
Meaning silencing both together had a greater effect than just silencing one or the other alone.
Now, that's a key insight, isn't it? It's not just hitting 2 targets. It's that doing so creates a sort of synergistic benefit, offering potentially a more powerful way to calm down that airway inflammation.
That's exactly it. And they saw evidence for this greater reductions in those key type 2 messengers, the cytokines IL-5 and IL-13.
Okay.
A less activity from those allergy-related eosinophils, lower protein levels in the lung fluid. It all points to a significant reduction in inflammation. They even saw hints of reduced lung scarring or fibrosis.
Wow.
Exactly. And if we connect this to the bigger picture, a multi-target inhaled RNAi like this could really simplify treatment. Imagine maybe one inhaler addressing multiple drivers of inflammation right at the source.
That could make it much easier for patients to stick to their treatment.
Absolutely. And it could be a real breakthrough for those with severe asthma or, those who don't respond so well to the current single-target biologic drugs.
It makes a lot of sense. If you can target multiple key instigators early on, you might get a more comprehensive effect. Now, the sources also dig into RAGE. That's RAGE, receptor for advanced glycation end products. That sounds like another interesting target.
It really is. RAGE is a type of receptor on cells, like a docking station, if you will. It recognizes a really wide variety of danger signals.
Like what kind of signals?
What molecules released by damaged cells, bits of microbes, even those advanced glycation end products it's named after, which are byproducts of metabolism.
Okay.
And when RAGE gets activated by these signals, it really amplifies inflammation. It does this by turning on key signaling pathways inside the cell, like NF-key, which is a major inflammation regulator.
And it's common in the lungs.
What molecules released by damaged cells, bits of microbes, even those advanced glycation end products it's named after, which are byproducts of metabolism.
Okay. So RAGE is like a central alarm system on lung cells then, triggered by a whole bunch of different threats, all leading to more inflammation.
That's a great analogy, yes. So the LMJ 44 paper discusses a preclinical study using mice. These mice were genetically engineered to have only one working copy of the RAGE gene. They would call this RAGE haplodeficiency.
Okay, so less RAGE activity.
Exactly. And they looked at these mice in 3 different models of asthma, each triggered differently. One by a fungal allergen, Alternaria, one by house dust mite, a common one, and one even by that IL-33 alarmin we just discussed.
And the fact that they saw consistent results across 3 different triggers, I mean, that really suggests RAGE is playing a fundamental role, doesn't it?
Absolutely, that's the key finding. The mice with reduced RAGE consistently showed less airway inflammation in all 3 models.
Wow.
And importantly, this wasn't just limited to the typical allergic pathways. They also saw reductions in other inflammatory signals like type 17 cytokines. This suggests RAGE's influence isn't limited to just one type of asthma inflammation. It might hit both eosinophilic and neutrophilic types.
So RAGE seems to be involved in a much broader spectrum of asthma inflammation than just the classic allergy-driven type.
Precisely. They also observed lower levels of signals that attract monocytes and macrophages, other immune cells, and fewer signs of tissue damage overall. This really implies that RAGE could be a super important target across different asthma subtypes, maybe even offering a disease-modifying effect by acting so early in that inflammatory cascade.
This really could be a game changer for asthma therapy. A single target that could potentially help a wider range of patients, almost regardless of their specific inflammatory profile.
Okay, but the research didn't just stop at asthma, did it?
No, it didn't.
They also looked at AR-RAGE, that's Arrowhead's inhaled rage targeting siRNA in models relevant to COPD and ARDS. These are conditions where a different kind of inflammation, driven by neutrophils, is really significant.
Right, and this is where it gets, well, potentially very impactful, especially because we don't have many, or really any, effective targeted therapies for this neutrophilic inflammation in COPD and ARDS right now.
That's a major unmet need.
Huge. The sources highlight how these neutrophils, a type of white blood cell, they really contribute to the lung damage in both conditions, including through this process called netosis.
Yeah, where they release these sort of web-like structures made of DNA and proteins nets, which can actually trap pathogens, but also really fuel more inflammation and damage.
Okay, so quite destructive. And AR-RAGE was tested to see if it could counter that.
Yes. They evaluated it in rat models designed to mimic key aspects of these diseases. One used elastase to induce emphysema, like in COPD,
Right.
and the other used LPS, a bacterial component, to induce lung injury, modeling ARDS.
And the results.
In both models, inhaling AR-RAGE led to a significant reduction in the number of neutrophils getting into the lungs.
Okay, so fewer inflammatory cells arriving. That sounds like a crucial first step. What else did they see?
Well, they also saw less damage to that delicate barrier between the air sacs and the blood vessels, the alveolar capillary barrier. That's a critical problem in ARDS.
Definitely.
And reduced levels of enzymes called matrix metalloproteinases, or MMPs. These are enzymes that can chew up lung tissue in these diseases.
Just less tissue breakdown as well.
Exactly. And importantly, ARO-RAGE also suppressed key inflammatory messengers, cytokines like IL-6 and TNFR+, markers of that netosis process we mentioned, and even HMGB1, which is another one of those danger signals that can activate RAGE itself.
So it's a feedback loop. RAGE activation leads to signals that activate RAGE more.
It can certainly contribute, yeah. HMGB1 is one of those DAMPs, danger-associated molecular patterns, that RAGE recognizes.
Okay, so it seems ARO-RAGE isn't just cutting down neutrophil numbers, it's also dialing down the whole inflammatory environment they create in these really severe lung conditions. The LMJ44 paper even calls it a potential dual-purpose therapeutic for COPD and ARO-RAGE.
And that versatility is, I think, a really key takeaway here. ARO-RAGE seems to show potential across a wide spectrum of pulmonary inflammatory diseases, not just the type 2 high asthma we often focus on.
Right.
But also those non-eosinophilic and mixed inflammation types,
Yeah.
the kinds we see in COPD, ARDS, and even in some treatment-resistant asthma cases.
It's pretty remarkable, actually, how one target, RAGE, appears to be so central in these different diseases with, quite distinct underlying mechanisms sometimes.
It really is. It highlights how interconnected these pathways can be.
The sources also touch on the delivery methods and the progress towards human testing, right?
Exactly. The use of those heterodimeric siRNAs for IL-33 TSLP, that single molecule silencing 2 genes, that's quite innovative.
Yeah.
And also the use of animal models that have been, let's say, humanized engineered to have human versions of these target genes. That really strengthens the translational relevance of this research for potential human therapies. We're getting closer to seeing how these might actually perform in patients.
And AR-RAGE itself, it's already in early-stage clinical trials for asthma. I think I read the initial findings showed significant target gene knockdown and a decent safety profile.
That's right. Data looked promising, good target engagement, well-tolerated. They even suggested potential for less frequent dosing down the line based on the duration of effect.
And the platform they use, this TRiM platform, that's key for getting it delivered properly.
Very key. The TRiM platform from Arrowhead is specifically designed to improve delivery of these RNAi molecules right to the cells lining the airways, deep in the lungs.
Okay.
This helps overcome some big challenges with inhaled therapies generally, like getting the drug to the right spot, making sure it's stable once it's there, minimizing off-target effects elsewhere in the body. It's quite sophisticated. They're also looking at subcutaneous injection under the skin as another potential route.
Right. So while there are definitely still hurdles, refining delivery, fully understanding long-term effects, the preclinical data across these different targets and disease models, it looks incredibly promising, doesn't it?
It really does. The next crucial steps, obviously, involve continuing those clinical trials. Being smart about selecting patient populations who might benefit most, perhaps using biomarkers.
Right. Stratifying patients.
Exactly, and really rigorously evaluating clinical outcomes, how patients actually feel and function alongside those inflammatory markers in human studies, and exploring combinations, too, how these might work with existing treatments.
Okay, so just to sort of wrap this up, we've seen really compelling preclinical evidence that inhaled RNAi targeting both IL-33 and TSLP can have significant, maybe even additive, anti-inflammatory effects in asthma models.
Yes.
And the research on RAGE, that's particularly striking, showing its broad involvement in driving both allergic and neutrophilic inflammation across models of asthma, COPD, and ARDS.
And we shouldn't forget those innovative delivery strategies like the heterodimeric RNAi and the encouraging early clinical signals for airregular asthma. It all points towards a future where maybe inhaled RNAi could provide more targeted and potentially much more effective treatments for a whole range of these inflammatory lung diseases.
Addressing both the eosinophilic side and the neutrophilic side.
Potentially, yes,
Okay.
which would be a major step forward.
This has been a really insightful deep dive. Considering how interconnected all these inflammatory pathways clearly are in lung diseases, it really makes you wonder, could a multi-target approach or a key upstream target approach, like with RAGE using inhaled RNAi, offer a more fundamental way to manage these conditions, compared to our current treatments that often hit just one target or mainly manage symptoms?
It's a critical question.
And I suppose, what are the biggest remaining hurdles, scientific, clinical, that we still need to overcome to really unlock the full potential of this platform for patients? Some really important questions to keep thinking about. Thanks for joining us.